So, and you're exactly right about the BDNF and all these other different growth factors and the plasticity. But anyway, so insulin resistance is one of those factors that you can see early on in Alzheimer's, and some people call it brain diabetes or whatever. A second one is that the neurons seem to have some dysfunction of their mitochondria, the little energy factories that make ATP, and ATP levels fall early. There's this mitochondrial dysfunction, and there's inflammation. And so these three things: neural inflammation, mitochondrial dysfunction, and insulin resistance. And you know, when I was studying fructose in animals, I was showing that they induce insulin resistance systemically, and that they induce mitochondrial dysfunction, reduce the ATP levels and suppress the mitochondria, and they cause inflammation. I go, wait, this is the same biosignature as what's going on in the brain. Could it be true that fructose could be doing this? And what's very interesting is that if you eat fructose or sugar, most of it is removed in the liver, and only a tiny bit gets to the brain. So it seemed like there was some paradox. There was some problem. Why is it that very little fructose gets to the brain if fructose is driving the disease in the brain, like what I was thinking? And then we had these really cool, not so cool discoveries, Robert, that the body can make fructose, and that you don't have to necessarily eat fructose. But when you eat sugar, the body makes fructose. So the body makes fructose in response to sugar, and the body makes fructose in response to high glycemic carbs like potatoes and rice. And it also makes it in response to salty foods. And all three pathways is due to a particular enzymatic reaction that I call the polyol pathway. But what it is glucose can be converted to fructose. And so when you eat a lot of high glycemic carbs, the glucose levels go up in the blood. That's why CGM is so helpful. That's why levels is so important group, because they provide these CGMs. But when the glucose goes up in the blood, fructose starts to be produced in the brain. And this has even been shown by a group at Yale in humans, that if you raise blood glucose, fructose levels go up in the brain in humans after about 40 minutes. And it's very significant. And we found that high glycemic carbs and salty foods, salt actually triggers that reaction of glucose to fructose. So there are these foods that we eat that can generate fructose. And when it goes up in the brain as well as in other tissues. So this led me to think, well, perhaps what's going on is we're eating foods that are raising fructose levels in the brain. And then I started looking at it, and I realized that there are all these data that show that, sugar intake is a risk factor for Alzheimer's. It's a risk factor for cerebral atrophy. High glycemic carbs are a risk factor. Salty foods are a risk factor. All the things that generate fructose are risk factors for Alzheimer's, and obesity and diabetes, which are signatures for fructose production or intake, they also are risk factors. So then we had this connection between the risk factors for Alzheimer's are the same risk factors for raising fructose in the brain. You give fructose to a starving animal, it will be converted to glucose because it wants fuel, immediate fuel. But if you give fructose to a fed animal, it will lower the ATP and activate this process that leads to obesity and metabolic syndrome. And it's really interesting that animals will eat fructose to prepare for times when there's no food around. Like the bear will eat fructose in the fall in the berries. It'll eat thousands of berries. It doesn't eat like one natural fruit. It'll eat thousands, and it will activate this switch, and it activates it by dropping the ATP in the reduces mitochondrial production of ATP. It induces insulin resistance and all these things as a mechanism to aid survival. Well, one of the really cool things that we have kind of discovered, or at least have really looked at carefully with others, is that fructose stimulates foraging. And so when fructose goes up in the brain, for example, or when you give sugar, you will stimulate a foraging response. And that foraging response turns out to be very important in Alzheimer's, because the way that you stimulate foraging, to forage, you have to go into an area that you've not been. You're looking for food. You have to be willing to go into areas where you've not been, search for food, look around. You have to look quickly. You can't spend a lot of time. You can't deliberate. You can't have a lot of self-control. You need to just be able to plunge in and do it, get the food, get out. It turns out that to do this foraging, you actually have to suppress certain areas of the brain. Like the cortex is really involved in self-control, like this, especially the frontal cortex. And so when you give fructose, you inhibit the activity of that area so that you have less self-control. It's sort of like alcohol, actually. Likewise, you'll if you give fructose, you'll inhibit the area for recent memory, because the recent memory is, you don't want to really have vivid memories of how dangerous it is, where you're going to go, right? And it will also like stimulate impulsivity and all these things. And the way it does it, it works on certain regions of the brain. And guess what? Those are the insulin dependent regions of the brain. And certain areas of the brain it stimulates, like the anterior cingulate, for example, is a part of the brain that's important for foraging, and fructose stimulates that, but other areas it inhibits. And then when you look at that, you find this amazing thing. Alzheimer's affects the areas of the brain that are inhibited by fructose, and the areas that are stimulated by fructose are preserved. So like the occipital cortex, so that you can see the food, it's not affected very much in Alzheimer's. The anterior cingulate, which drives the foraging, that's not really affected in Alzheimer's. But the cerebral cortex, the hippocampus, the entorhinal cortex, all these different areas that are inhibited by fructose are actually the signature of where it occurs. And it's the same areas where insulin resistance occurs. To kind of wrap our hypothesis together, basically, if you give sugar to animals, which has fructose in it, after about 8 weeks, they have trouble walking through a maze. They have, normally they can get through a maze, and as the more times they go through it, the faster they go. If you give them fructose, when they go through the maze, they don't get faster. They continue to have trouble getting through the maze. Then, if you look in their brains, you find insulin resistance, you find mitochondrial dysfunction, you find a drop in BDNF, your nerve growth factor, you find a drop in ATP, mitochondrial dysfunction. It is exactly what we're talking about. And then if you go out to 16 weeks, they actually start developing amyloid plaques and tau protein. You and I both got to the same place. A drop in ATP is probably the key issue driving Alzheimer's. And there's only one nutrient. There's only one nutrient that lowers ATP in the cell, and that's fructose. And fructose levels are high in patients with Alzheimer's, 5 to 6-fold higher than in normal age-matched controls. It's amazing how complicated fructose is, but how it all leads in excess leads to morbidity. And, as we get towards the end of the hour, it's really fun talking to you about how we can learn more and more about what fructose does and how it probably has roles in not just obesity and metabolic syndrome, but neurologic diseases and ADHD and bipolar disease and Alzheimer's and seizures and GI illnesses and anaphylaxis. It's such a major driver. It's a big animal. Fructose is a big animal that does a lot of things to a lot of systems. I know you have this interest in fructose and cancer and fructose and alcoholism, and we've also linked fructose as driving the Warburg effect, for example, which is fructose is the perfect fuel for cancer cells, and fructose is involved in alcoholism too. So, we need to have another session to dive in more. but because it's sort of scary, you know. I was when I started seeing that fructose could be involved in so many processes, I looked in the mirror. I said, am I tricking myself? There's like too many diseases that fructose is important. And then, but you know, then you do the studies and you block it or whatever. And it, you know, it seems like it's more like we've discovered you know, a really important pathway. And I think the reason is because fructose is the only nutrient that lowers the energy in a cell. Most foods, when you eat it, you increase the ATP and the excess can go into fat. In fructose, you drop the ATP, so the energy that comes in is go to maintain energy balance goes into the fat directly. And so you maintain a low ATP, but high fat program. And that's what people with metabolic syndrome have. That's what people with diabetes have. That's what people with Alzheimer's have, you know? And so we're looking at a signature.